Perfect. Hello, everyone. Welcome. Thank you for joining us today for our March session of the Winter Lunch and Learn series brought to you by the External Committee at the Graduate College. My name is Mickey, and I will be the moderator for this afternoon's session. We are very excited for two of our scholars to have the opportunity to share their presentation with you today. So just to give a brief summary and outline of what's going to happen today, we will start off with some opening remarks, and then I will introduce our presenters. They will have about 20 minutes to share their presentation with us. And at the end of this session, so after both presentations, there will be a Q&A period. Please use the Q&A box to enter your questions at any time during their presentation. So you don't have to wait until the very end to do so. So if you have a question that you don't want to forget, you can easily write it in the Q&A and we can address it when it comes to the actual Q&A session. And finally, after the Q&A session, we will conclude our session with a short summary. So before we get any further, I would like to take this opportunity to acknowledge the traditional territories of the people of the Treaty 7 region in southern Alberta, which includes the Blackfoot Confederacy, comprising the Siksika, Piikani, and Kainai First Nations, as well as the Tsuut'ina First Nations and the Stoney Nakoda, including the Chiniki, Bearspaw, and the Wesley First Nations. The city of Calgary is also home to Métis Nation of Alberta, Region 3. I would also like to note that although we are connecting virtually today, the University of Calgary is physically situated on land adjacent to where the Bow River meets the Elbow River, and that the traditional Blackfoot name of this place is Mokinstisi, excuse my pronunciation, which we now call the city of Calgary. So just to give a brief overview of what the Lunch and Learn series itself is. So the Lunch and Learn series is a public presentation series that aims to bridge the graduate college with the Calgary community. Our series has been ongoing since April 2020, and we are continuing the series into this fall. Currently, that's the plan, I think. But yeah, during each Lunch and Learn presentation, the graduate scholars will present their diverse research using language that's appropriate for the public. So without further ado, today we have two speakers. Our first speaker is Tanille Shandro, who is a PhD student in the Department of Chemistry at the Faculty of Science here at the University of Calgary. Their research focuses on developing novel biosensors to enhance disease detection. Their presentation today will be on Gout Biosensors, developing a molecular level medicine diagnostic for crystal arthropathies. Thereafter, Frank Lee, who is a PhD candidate in the Department of Community Health Sciences at the Cummings School of Medicine, will share with us a presentation on health data science and electronic medical records data. Frank's research focuses on developing electronic health records-based case phenotypes and utilizing them for risk adjustment analysis. Okay, Tanille, the floor is all yours, so I will stop sharing my screen and let you do so that you can go ahead and get started.
Awesome, thanks so much, Mickey. Great. Okay. Awesome. Thanks so much, Mickey, for the great introduction. I'm really honored to be here today, and it's just a pleasure to be invited to talk about my research as part of the Lunch and Learn series. Many of you that I recognize in the participation bar there are I would know from different capacities here at the university. So I am also the Graduate Student Association president, as well as working as a community ambassador in Crow's Nest Hall, so our single student housing here on campus. And so it's such a great experience to get to talk about my research to all of you, because these conversations are typically quite different when I'm in those kind of positions. So today I'm here to talk a little bit about my research project and hopefully get you all a little bit more caught up on your biotechnology knowledge, but I will keep it kind of a light lunch learning session so that we're not diving into the weeds and getting lost. So as Mickey introduced, my project is called a Gout Biosensor, developing a molecular level medicine diagnostic for crystal arthropathies. And I promise we'll unpack all of this jargon language throughout the presentation so that you feel a lot more comfortable looking at a lot of these words. So first off, we have to address kind of what a biosensor is. So a biosensor, although it's not a very inviting term that I feel everyone knows they know what it is, it's really just a technological device that finds something with a biological molecule within a sample. And they are kind of in our everyday lives, depending on who you are. And so a good example of a biosensor is a blood glucose monitor, which does exactly that, monitors blood glucose levels for individuals with diabetes, as well as things like pregnancy tests. And these monitor a type of protein that are present when you're pregnant. And so these essentially take biological molecules and tell you if you have them in your system or the quantity that you have in your system. And so what my project really looks at is gout and how to develop a better system to detect and then create a treatment for gout. So gout is a painful joint disorder that's very common in our current population, but it is curable when properly diagnosed. And one of the problems that we currently have is that proper diagnosis. And so that is really the focus of what this is. So I'm really excited to discuss with you kind of why this is important and how we're doing it. So in terms of gout, the basic knowledge is that gout is a joint disease that affects the metatarsal joint of the foot. And really that's just a fancy term for this big first joint of the big toe. And what happens is there's a deposit of what's called monosodium urate crystals, or MSU crystals. And there's these long rod-like structures that build up in the joint in areas of low blood flow. And what happens is they deposit in this joint, and when the joint is disturbed, they're knocked off of kind of these deposition areas, and they are then accessible to be attacked by these macrophages as part of your inflammatory response. And so once the macrophages have access to them, you'll see swelling and inflammation. And so if you've ever stubbed your toe, you know what that feels like and how awful it is. But really, when you have gout, it's 1,000 times worse. And people who suffer from gout really describe it as a very unpleasurable experience, being under what's called a gouty attack, which is kind of those symptoms that people feel of when they're experiencing gout. So how do these crystals form? So essentially, there's the basic building blocks of DNA. So you have the top category, which is pyrimidines, and those are 1-ring structures, and then purines are the 2-ring structures at the bottom there. And these you consume through your daily diet. And as part of purine breakdown within your body, you get a compound called uric acid. And this uric acid, you're literally probably breaking down your breakfast or lunch at this time already by now and creating this, but your body's really good at excreting it, and it's excreted through your urine. And so there's already a system in place to, take care of all of this within your body. So the issue really becomes when you have too much in your body. So the uric acid compound in the physiological pH, so sitting within your blood, becomes a urate ion. And that urate ion then has the ability to interact with the sodium ion. And these two collaborate together to create these really unpleasant rod-like crystals. Now, this condition is known as hyperuricemia. And if you really break down the kind of basis of this word, hyper is too much uric acid in the blood. And so this is a common condition when individuals are unable to properly address the uric acid in their blood. And the uric acid has the ability to stack up on one another to create these long rod-like structures. They grow in terms of crystal growth, or it's called nucleation again, which is just a fancy word for growth. It grows in one direction faster than it grows in the other. And that creates this long rod-like structure that you see on the right-hand side there. So to kind of get an analogy of what this looks like and how it's treatable is, I like to imagine the human body as a sink, and the water being that uric acid buildup. So how gout forms is when there's potentially too much water in the sink. And this can come from your consumption in your diet. So it comes from fish, from red meats. It can come from high fructose syrups, so such as sodas, as well as other diseases such as obesity. Now, another common factor of having too much uric acid in your blood is when your body can't get rid of it. So if you're like putting a plug in that sink, and this can happen if you have alcohol or if you have kidney failures or kidney diseases that prevent you from processing purines. And so it's an unpleasant situation, but it is curable. And typically this in previous years, way back when, they used to actually call gout the disease of kings, because gout, this kind of lifestyle of leisurely eating and alcohol, was only accessible by kings. But now it's much more accessible. As you all know, you can kind of head around the corner to the grocery store and liquor store and get all of these purines ready for your diet. But in order to address it, there's kind of two ways that you can cure it. So you can either stop the water, which includes taking these kind of things out of your diet, as well as changing your lifestyle to include exercise, or taking out the plug, which would be kind of eliminating alcohol from your diet, as well as taking some drugs to help your kidneys process purines. So kind of the summary up until this point, as I know science might not be everyone's cup of tea, is that uric acid is caused by uric acid causes hyperuricemia that forms MSU crystals, which then causes painful symptoms of arthritis, or what's called another fancy word for a joint disease, which is crystal arthropathies. So joint diseases that are caused by crystals. Now, crystal arthropathies are quite common. So we have the gout one, which we just talked about. So MSU crystals causing gout, but there's also lots of other crystal arthropathies or crystal joint disorders, such as one called pseudogout. Now, pseudogout, as the name suggests, presents itself very similarly to gout, and it has the same symptoms and kind of same everything except for the type of crystal that forms it. So the type of crystal that forms pseudogout is calcium pyrophosphate crystals. And so very commonly, pseudogout is misdiagnosed as gout because of the symptoms and the current diagnostic methods. And then it's improperly treated because we're not addressing the proper chemical makeup of the crystal within the joint. So the current detection strategies is currently called compensated polarized light microscopy, for any of you that are interested in kind of diving down that hole further. But this is kind of a typical situation that they would see. So what the current detection strategies look at is they take a sample from this infected joint, then they look at it under a special microscope and see something similar, where they're looking at the shape of the crystal as well as what's called the birefringence. So kind of a special property that shows different crystals, specifically MSU and CPP, as different colors. But as you can see here, it's kind of just a situation of crystal soup. And so it's poorly sensitive to say what kind of crystals are in that sample. It's operator dependent. So it really depends on which doctor essentially gets the sample and looks at it with their own two eyes. So you versus the person sitting next to you might say two different diagnoses. And so that creates a situation where it's often inaccurate in detecting gout. So what we're trying to do is essentially create a method where you can detect this without the human eye using peptide engineering. And so what this ideally would look like, and this is where it's sad that it's on a Zoom webinar, because I would love to hear the oohs and ahs of my PowerPoint animation skills. So here's what we're looking at in a very boiled down form. We're looking at creating a system where you can take that fluid from that affected joint with potential crystals in it, put it in a sample with an engineered peptide that only binds specifically to the MSU crystals. And to be able to visualize that, we need to have some sort of visual component. So on these peptides, we add a fluorescent molecule. So when it's under green fluorescent wavelength, it'll glow green. So if we add that into a sample with the MSU crystal and let it incubate for a little bit, we'll see the sample glow green. And kind of vice versa, if there was no MSU crystals, the peptides wouldn't be able to bind to the crystal. It would be washed off, and then you would get no green glow. So really a clear way of determining what kind of, at the microscopic level, what crystal you're working with, rather than using the human eye. So really the big question is, how do you design these peptides? This is what I work on. So there's kind of four main structures of proteins. So the most basic structure is amino acid strands, and that's kind of on the far left here, where it's really depicted in a cartoon way of being kind of like beads on a string. And then when you move up in protein complexity, you start with secondary structure, where you have kind of a helical structure, as well as beta sheets, which is a different type of structure. And then it kind of builds off of that. So it creates a larger protein, and then those proteins can interact with one another to create this complex protein molecule. So when we're looking at doing biosensor technology, we're really looking at that smaller end. So the amino acid structure that creates the secondary structure, kind of hand in hand, working at that lower level. So how we do this is through a technique called phage display. And what phage display is essentially a way to expose different amino acid strands to the crystal or your target of interest. So what we do is we take a bacteriophage, which looks like this. So kind of just a fancy vehicle to be able to introduce the peptide strands. And we attach peptide strands to this bacteriophage to be able to introduce it to the crystal. Now you have a whole kind of pool, it's called a phage library, where you have these bacteriophages, but they all have different peptides on the end so that you can see which one would work the best. So you expose these to a crystal and go through a series of washes and amplification, so growing up which ones bind the best. And you end up with just one, ideally. And then you would take a deeper dive look into what that amino acid sequence is to see what those chemistry properties are that are interacting with the crystal at a very molecular level. Now, something that our research showed that was very interesting and unexpected was that these M13 bacteriophages with no peptide on the end were interacting strongly with the crystal themselves. So it really didn't have to do with the amino acids that we were exposing it to, it was the M13. And so when you look at this structure, you can see it has five proteins, but one major coat protein, which is this black coat protein. And it's easy with this kind of cartoon characterization of it to not envision these also as peptides or also as protein structures, but they truly are. It's a coat protein. And so when you break that down even tinier and look at its structure, this is what one coat protein looks like. So the coat protein is made up of 50 amino acids, so kind of like 50 beads on a string, and it creates an alpha helical structure, which is essentially just a fancy spiral. Now, looking at this, 50 amino acids is kind of too long to expose to the to the crystal to really understand which component is interacting best with the crystal. So what we've done is cut this up into tiny little strands of 15 amino acids. And we then took those little chunks and put on that fluorescent tag to be able to optically visualize it. And when it's under green fluorescence, to be able to see something, because otherwise it'd be very anticlimactic where you wouldn't be able to tell what's going on at a molecular level. So then what was done was these were taken and incubated with an MSU crystal, all individually, to see which kind of chunk of that 50 amino acid strand has the best, strongest, most specific interaction with the MSU crystals. Now, this is what kind of typically looks like under the microscope that I work with. And so these are all the crystals under the microscope. And when we have a sample of a peptide strand that doesn't interact with the crystals, what you see when you put it under green fluorescence is just a black screen, nothing super exciting or fancy. But when you look at a sample that has a proper kind of a better interaction between that crystal and that engineered peptide, we see this green glow. So here you can see one of the peptides that we're working with actually has some interesting, convincing situation where we're kind of looking into further what that looks like and what that interaction is at a molecular level. So there's still a lot of work to do, and it's a very exciting project to be on. And some of the promising results definitely keep you optimistic about what's happening. But really the direction that I'm excited to go in is right now what the project is looking at is working on synthetic MSU crystals, or crystals that I create in the lab. And kind of the direction that this is going and needs, we're almost there, which is exciting, is working on human samples. And so really taking that proof of concept created with the synthetic crystals and interacting it with the human samples to be able to see if we can more accurately and selectively detect MSU crystals within a gout sample. So thank you so much for attending today. I really appreciate you taking your lunch time. I know we were chatting just before everyone got on that there's a lot of Zoom meetings and a lot of online time right now. So I really appreciate you taking your time to come and listen to what I work on in the lab every day. And I'm happy to answer any questions at the Q&A period, but I'll hand it back over to Mickey to introduce Frank.
Once again, thank you, Tanille, for that wonderful presentation. And if it's an opportunity at least to appreciate it, you get the oohs and ahs for your animations from me. Thank you very much for your presentation. Just a reminder to our attendees, if you do have questions for Tanille, just reiterating to use the Q&A box on the Zoom functionality, and we will go through the questions during the Q&A period. And now, Frank, you can go ahead whenever you are ready.
Sounds good. So just quickly share my screen. So thanks, Tanille, for that great presentation. And hello, everyone. As Mickey have introduced, my name is Frank Lee, and I am a PhD candidate in Department of Community Health Sciences. I also hold a senior analyst position in Alberta Health Services in their data and analytics team, as well as a support unit for other data-related inquiries on the provincial data sets. And I am located in the Center for Health Informatics. So my talk is gonna go a little bit different direction compared to Tanille's. And just even from the title, there's a lot of jargons in here. So I was thinking about this after external committee approached me about doing this talk. And I consulted my supervisor about this, Dr. Huda Kwan and Dr. Jun Lee, they're my supervisors. And they suggested that that we talked, and the decision was to go a little bit broad. So that's what I would do, but I will still show some of the slides showing some of the technical details. But if you have any questions about any technicals and insider, a lot more detailed information, feel free to send me an email or email to the CHI, or we can take it offline for discussions.
Before I want to start. Oh, sorry, Frank. Before we continue any further, I just wanted to let you know that we're seeing your presenter mode and not the slides themselves. If you wanted to change the presentation so that.
Hold on, presenter view.
So it might just be sharing the one screen inappropriately.
Yeah, because I put 3 screens on here set up here, so that might be why.
For sure.
It's okay. I mean, if you guys are okay with it, if you guys are okay with it, like there's no notes in any of these slides, so.
Perfect. Then I don't think there's any issues. For the sake of time.
I have no issues.
You're more than welcome.
Yeah, I have no issues here.
Perfect.
So sorry about that. Sorry to everyone.
Sorry for interrupting you there.
Yeah. Oh, actually, this might work. Okay. So I want to start with the concept called this concept called learning health systems. So this is a framework that is currently operational in many of the health systems across the world that is talking about how do we transform the data that is collected in the health system onto improving our clinical care practices as well as quality of care. And there are sort of 3 arms to this learning health system. The first one is the sort of called the data to analytics component, where health systems collect a lot of data. Like and I'm sure all of us have had interaction with health system at one point. And just even them asking us questions on, okay, what is your age? Do you have a specific underlying conditions? All those informations gets recorded and they sit in the system. And the second arm of the learning health system is sort of getting at, taking that data to the analytics, and from analytics onto clinical practices. And from there, so basically that whole second arm of going from data, so analyzing the data that is collected in the system into some form of analyzed data products, which becomes the information, useful information that can be used to improve healthcare. And then the last term is really getting at the knowledge translation. What do you do with that information that you have collected? So for my thesis and for basically in the Center for Health Informatics, the slide that I'm showing you here is how we have decided to tackle the sort of what we call the pipeline to learning health system. So the idea basis is that, in our health system, we have a lot of data. a good example, there's electronic medical records data, which is collected at all of in hospitals. There's a lab data, and AHS definitely has a lab database that's standalone on its own. On top of it, additionally, we also have HealthLink data, the 811 calls, and that have become really important tool as we are in the middle of a pandemic. So our sort of take on is we have all these different data sets, and what we're going to do is through research and collaboration, we will transform that data by first link the data and then follow up with developing terminologies and algorithms to analyze those data, the collected data, and also assess data quality. The transformed data would then lead into some form of analytics. This gets at the entire concept of modeling and visualization. And visualization is a key component for us because it helps us delivering the information to it helps us, they deliver the crucial information to the layperson who may not have a knowledge in all of the technical details that we may be undertaking. And then our hope is that this will eventually lead toward the precision medicine and precision public health, which sort of gets at the goal of, tailoring the treatments and approaches to each specific individual. And through this entire process of the outline here, one of our aim is to really develop the training and capacity associated. And so the question sort of became, well, how are you gonna, so you have the framework that you're operating by, how are you going to analyze all the data and what kind of techniques will you be using? So that's where this whole idea or the field of health data science comes in. So health data science is a branch of data science that actually combines epidemiology and biostatistics onto the emerging discipline of data science that's currently ongoing across the world. And some of you in the audience may know, but epidemiology, which sort of studies the distribution or how the disease is spread in the population, along with studies design and topic matter expertise, and biostatistics, which sort of gets at the entire concept of statistical modeling and theories. These two techniques have been existed since 18th, 19th century. And the really first good example of epi and biostats sort of being used is the cholera outbreak in London, the investigation by Dr. John Snow, who actually visualized cases onto the map and identified a one pump relay was responsible for causing that cholera outbreak. So there was a so these two disciplines have emerged as sort of what we call conventional methodologies in health analytics. Now, the data science is a new framework, sort of the emerging framework that comes in, especially with and really focuses on the artificial intelligence techniques. So these are machine learning techniques and natural language processing, which is sort of aiming for, giving the computers the ability to read a language as if they're talking to a human. Then the data science component also has a fair bit of data management and engineering principles added onto it. And to achieve our objectives in using electronic medical records data, like the data science techniques and the basic epidemiology biostatistics, the combination of three is actually crucial for us. So on to this thesis topic. So as I mentioned before, my thesis topic was looking at basically is taking the electronic medical records data through that framework that I showed you earlier. So the first step for us, for me, myself, and a good number of my team members were first, okay, we need to figure out what we are working with. So this first paper that we published, Unlocking the Potential of Electronic Health Records for Health Research, is basically described, the whole entire purpose of this paper was describe the Sunline's clinical manager for research purposes. And in this paper, we do discuss the types of data that is contained in the SCM. So there are what we call structured data. So these are the data where the format is sort of the placed upon. So these could be gender, could be coded as male or female. It could be admission date, which is coded as a specific date format, right? And then there's a free text data, which are just like, the free text narratives that doctors write in their clinical notes as they work with patients or as they provide care to the patients. Then this paper also discussed advantages and challenges, basically, of using EMR data in health research context, taking account of some of the epidemiology and biostatistics concept. And once we sort of understood what this database was working on, and one thing that we also did in this paper is that we actually showed the flow of data from from the point it was collected all the way to the point where it ends up in the databases. So basically, doctors and nurses and other health providers, they would write their notes or enter information from what we call the front end at a console in the hospital. And then, without going into too much details, basically all that data becomes transferred over to multiple databases and eventually ends up in the data warehouse in the Avatar Health services. And understanding of this structure was crucial for us because for some of the projects that we're working on right now, we actually have to pull the data from these databases. And understanding the structure of these databases led to this work and this paper that we published in Canadian Journal of Cardiology Open. And so this database called CREATE, and this stands for Clinical Registry, Electronic Medical Records Data, and Administrative Data. And what we have effectively done is basically link those multiple types of databases onto a single database. And here, again, I don't want to go bore you with too much technical details, but what we have done was we were successful in linking about 31,000 patients and divide them into two different types of cohort, one in inpatient cohort and the outpatient cohort, using some of the data science techniques and epidemiology techniques for selecting a specific population. And now the most important aspect for us was this is sort of the pilot project where we undertook the linking the SCM EMR data with other types of databases. And essentially, at the end of this project, we ended up with a mechanism for constructing the various types of databases. So we have right now, as presently, we have we have a good number of databases that's under construction or operational to answer specific research questions. So if you sort of recall to the framework that I was discussing, so we got the data linked together. And now the question becomes, well, what do you do with that linked database? So one of the aim of our team, and as well as my thesis, is sort of looking at this concept called EMR phenotyping. And this is basically getting at the simplest way to put it is how do you define conditions or events using specifically the EMR data? And once we sort of had what we were aiming for, in particularly the phenotyping area, we sort of needed to get a picture of what is out there. So we went on to do this literature review of chronic conditions specifically just for this particular paper's purpose, and went on to identify all different types of papers that were published across the world in the past 20 years. And at the end of it, we ended up with about 274 papers in it, accounting for 299 different algorithms of how others in the world have used to define chronic conditions using EMR data. So this literature review revealed a fair number of information to us. First, it told us that healthcare is a very unique environment. You may be dealing with the same disease, but people across the world have adopted different case definitions and have different way of interpreting the information. So in this paper, WUSAC does discuss some of the suggestions and recommendations for how to develop the phenotyping for those who are interested in it. But for our purposes, it gave us a really good picture of what is out there. And we also noted that this type of information is needed for building support system in health services. So here is so once we sort of had a picture of what we were dealing with, our team and now have moved on to developing basically the phenotyping some of the chronic conditions that we have. So this is a project led by Shanzhu. He is a faculty in the Center for Health Informatics. And the entire purpose of this paper was basically defined case definition for heart failure using EMR data and compared to the existing mechanisms. And this table here just shows you various different algorithms that were developed for this particular project. And again, without going into too much technical details, basically the whole point of what we assessed was we looked at the performance of varying data types and as well as the techniques, such as natural language processing and machine learning. And just to point out, here in this table, number 4, machine learning algorithm is written here. That is actually a algorithm that we developed from another project. So this diagram actually shows you the steps that were involved in developing that particular ML algorithm here. So this was another project led by Elliot Martin, who is another analyst on our team. And basically the whole aspect of this diagram is basically indicating that we are extracting those from the EMR system. We applied natural language processing, then we applied machine learning technique model for fitting a classification model. So in this case, it's called CART, which I think stands for classification and regression tree model. And basically we use that to sort of analyze for this project, specific hypertension, but also the same technique we have used for analyzing the heart failure data. So that is sort of where we are at in terms of our work. My thesis is definitely on EMR phenotyping, as well as some of the natural language processing models. And so we do have other projects going on in prediction models, developing NLP models. And right now we do have a good number of projects working with the various clinical and then health system disciplines. So we are actually engaged with hepatology, cardiology, oncology, and patient safety divisions for some of our projects right now. And our eventual plan is that all of this work that we develop, techniques and algorithms that we develop, will be used for surveillance and for building support system tools within health services. Now, in terms of the future directions, some of you may be aware that there is a large project called Avatar Health Services Connect Care. And that is basically building a province-wide clinical information system. So from our perspective, Sunrise Clinical Manager EMR is a citywide EMR system. So we are right now in a transition phase of going from citywide to the province-wide system. So a lot of our work will most likely be translated over to Connect Care, and then will help with Avatar AHS with some of their analytics. So going back to this picture, again, this picture does a great job at describing what my thesis and what the Center for Health Informatics is working on. We're sort of really have moved on from data access and linkage, and we have moved sort of at the level of data analytics right now. And so eventually we'll be focusing on some of the application component. So that's pretty much what I wanted to discuss. And I apologize for keeping it pretty high level, but again, there are many components and technical details that needs to be discussed. And if you have any questions or comments, feel free to contact me or the Center for Health Informatics. And I will pass it back to Mickey.
Oh, so once again, once again, thank you, Frank, for your presentation. I very much appreciated it. We had quite the diversity in the presentations today, so we will open the floor up for any questions for either Tanille or Frank now. For Zoom purposes and just for organizational purposes, please use the Q&A box. That's a part of the Zoom functions on the bottom of your Zoom program. And you can feel free to ask any questions related to their research, as well as those related to areas of their research if the presenters are comfortable answering them. We did get a couple questions that trickled in throughout the presentation. So I'll start with the first one that we got, which is directed at Tanille, which is, could you have tagged different colored peptides to see if the segments interact differently when you cut them down? So I assume this refers to your 15 base pair strands.
Sure. Awesome. Thanks so much, Mickey. Do you mind if I share a screen? I know that that kind of throws a wrench in the things. Just give me one sec, because whoever asked this, it's an anonymous person, but really you're on the right track. And I had this slide already from a different presentation. So you absolutely could do that in terms of like the smaller peptide strands. So the 15 mers is what they're called. But when you look at the chemical makeup of both the peptides as well as the crystal is slightly negative, and some of the peptides on different sections of that kind of long 50 mer are negative and some are positive. And so you absolutely could do that, but you would just see the black more likely on, like you wouldn't see anything if it was a negative interacting with a negative. But kind of you're exactly in the same mindset as me in terms of like, how can we make this better? So here's my dream, and it's called a multiplex bioassay. And essentially when you're looking at kind of what I was showing in terms of the crystal soup, where there's lots of crystals present, what the dream would be with this is creating an individual peptide engineered for each individual crystal, or kind of doing a sample stain. So you can see the yellow ones here are all different types, but you could stain them across the board if those are the ones you're not looking for. And then there's all kinds of techniques where you can look at and say like, okay, this is exactly what's in the sample based off of those different wavelengths of coloring the different fluorescent molecules. So absolutely, using different colors of fluorescence is a huge way to enhance our detection methods with biosensors. Thanks so much, Mickey, for letting me share the screen.
Not at all. Thank you very much for brilliantly answering that question. Then I'm just going to press done. Continue sharing my screen. All right, so now we have another question for Frank. This is from Aksher, and the question is, what sort of general filtering method, if any, is used in health science related data before feeding them to the algorithm?
So the general filtering method depends on the data type that you are going to be using for your specific project, as well as the sort of the utilization needs of the users. So a good example that I can take as one type of data example is the numerical data. So lab data, for example, has a lot of data there. So there's a lot of numerical data. Now, one thing with EMR data, and this is a challenge that we have recognized working with EMR data, is that EMR data itself is not designed specifically for research purposes. The EMR data is collected as a result of providing care to the patient, which means if from research perspective, Ashkar, so you're probably thinking in the lab data, you're possibly thinking about, the missing data, right? In the lab data, right? Only because not everyone will get a specific lab test on the specific day you want it to be to answer your research question. So there are missing data. And again, and this is where some of the epidemiology concepts come in. And in epidemiology, we refer to these type of situations as selection bias, where the data where there's a bias in the methodology, in the way of collecting the data, and will sort of distort the real true association between whatever we're seeing. So there are a couple of techniques that you can use, but of course, in terms of when you're working with that type of data, you need to sort of cut the data down. You cannot analyze the entire data set on its own. You do have to do some pre-processing in the data, and then fit the techniques as needed, because you may be potentially be seeing class imbalance in your data sets as well. So some of the general techniques in data science is still being used, but you have to take consideration of the data types in there. So I talked briefly about numerical data. The other type is the free text data. Free text data is much more trickier to work with, because first you have to convert all the data into a analyzable format. And in medical language, it can get tricky, because some doctors can say, oh, I suspect the patient has this particular disease, but no, I changed my mind. This patient does not have a disease, right? So these are kind of negations sentences that and these type of things are commonly seen. And so there are few tools out there that looks at analyzing this type of data. But for any free text data, you have to do, like, even there, you have to do a lot of cleaning the data. Spelling mistakes, a big one. The annotation issues is another one. So again, I don't want to get into too much detail because now we're getting too much into NLP, but those are some of the stuff that's involved when it comes to general filtering methods.
Awesome. Thank you for answering that question, Frank. I actually had a question to Tanille myself. Just when you were chatting about how you're hopeful to do human harvested, I don't know if that's the right word, but crystals from actual patients and such. I guess maybe because I'm not familiar with the process, but when you draw like liquid from the site of inflammation, is there an expectation that there needs to be like a minimum cutoff, and then it's just a yes or no if it's gout or not, or if it's the crystals or not, or is there like ways to increase the resolution and you can tell the severity or the amount of crystals based on the methods that you're working with?
Absolutely. Thanks so much for the question, Mickey. So yeah, in terms of working with human samples, there isn't an amount cutoff, I wouldn't say. Kind of what's different in this project versus some of the other graduate student projects that I know are going on with like human samples and human subjects. This is literally all done already in a biobank. So there's a partner doctor that we work with over at Foothills that has all of these samples already taken. And there essentially is gout samples as well as pseudo gout samples. And the pseudo gout is that CPP crystal. So ideally, once this proof of concept is kind of, troubleshooted, we would be able to introduce these peptides to both these samples. And we wouldn't actually, personally, I wouldn't know the answer, like what's gout, what's pseudo gout. It would kind of be matching up afterwards to see if the proof of concept is working. But in terms of like how much, or like almost like the concentration of crystals within a sample, the idea around that is really around what I'm also working on now. And again, I know Frank probably feels the same, but it's so hard to fit all this exciting stuff into 20 minutes. And so there's so many other things I'm working on in terms of automation. So really the images that I was showing are also still quite user dependent. And so looking at automation, that would be able to tell you kind of like, yeah, like this is how many fluorescent molecules you have within this sample. And that would kind of give you a better indication of what you're looking at in terms of concentration of a crystal in a sample.
Awesome. Thank you very much, Tanille, for answering that. We have another question for Frank from the Q&A. It's asking if you could elaborate on some of the opportunities and or challenges related to the Connect Care data.
So with Connect Care data, some of the interesting opportunity is that we are going to have a single EMR or clinical information system for the entire province. So by that, what I mean is any sort of AHS institutions is going to be using a single system. And that provides a very interesting opportunity because right now, very few places in the world, in Canada, probably not many, probably advertised maybe the only one in a few years down in the road, is the only province that's going to have such capacity. Now, so that's an opportunity, but it's also, that itself is a challenge because now we are looking at the entire province on its own, right? Like for example, the Edmonton already has Connect Care data implemented. The Connect Care system is already implemented in Edmonton. And in Calgary, that system is going to be online, going to be implemented pretty soon, I think. It's slated to happen sometime in this year. And opportunity-wise, like designing Chelsea, I know you're from my department, so you have a knowledge in epi. So you probably understand when we mean like we can literally do a population-based studies covering entire Alberta. That is like quite a bit of a, it's salivating, right? In terms of the things that you can do. But the challenge is also going to be, well, how do you then ascertain, knowing that the ConnectCare data is collected, based on providing care to the patient. And you probably have noticed that, each clinical unit may have their way of doing things. So even if you're measuring the same disease across multiple institutions, they may not be doing it the same way, right? So there's going to be some data quality challenges. And it's also the way the doctors write their notes in the clinical notes, or certain doctors interpreting specific lab tests. There's going to be a lot of variations. So I know the AHS is interested in applying some of these like integrated data analytics down in the long road, but I also think that's going to be a fair bit of interesting challenges there. Just trying to standardize the entire system that covers the entire province. I think that's going to take a lot more work. Of course, the vision is there, but that's definitely a challenge that we anticipate, I think.
Thank you, Frank, for answering that question. We probably have time for one more question if anybody is interested in submitting one on the chat. But if not, I guess I now have a question for Frank. This is, I guess, just out of personal interest, and maybe just because as humans are getting more advanced in the amount of information that's possible, and there's a lot of like privacy concerns when it comes to using technologies, but that translates over to the like medical side as well. Is there a lot of ethical dilemmas or concerns or potential challenges that you might kind of envision something so comprehensive like being able to conduct epidemiological study of all of Alberta kind of needs to take into consideration?
Right. So this is many of our studies are already facing some of these issues, but the privacy is one of the, because we do Alberta Health Services has to follow the Health Information Act that's provisioned by the Legislative Assembly of Alberta. So for specifically working with EMR data, the University of Calgary had to go through a fair bit of discussions on this topic with Alberta Health Services. And we do have a binding legal agreement signed between the legal councils of both organizations. And basically there's a fair bit of stipulations in there, which I think is, I will avoid going into detail, but one of the comment they made was like, we have to take the patient privacy as our uttermost priority. And for that reason, they asked us that at this point in time, those who are working with EMR data needs to have an affiliation with Alberta Health Services. That is one of the conditions that they placed on us, because the potential issue is with this area is the liability issue. If there's a massive data breach, right, then there's going to be a lot of consequences as a result of that. And so the university and the AH has had a fair bit of conversations surrounding that topic.
Right, it sounds more complicated than what I have to work with. Luckily, I get to work with non-humans, so privacy isn't as big of a concern. But yeah, no, thank you so much for answering that. Unless there's a very quick question, I don't think there's enough time for another question, unfortunately. So I'll leave the time for any quick question now, giving people the opportunity to type. But yeah, if not, those are all the questions that we had time for today. I wanted to once again thank both Frank and Tanille for the wonderful presentations today. We very much appreciated being able to hear about the research that you guys are conducting at the University of Calgary. And thank you as well to our guests who took time out of their day and your week to participate in the Q&A session, as well as participate in watching Frank and Tanille's presentations. Your questions were greatly appreciated. So it was a pleasure to have everyone here today. Hopefully everybody is making use of the wonderful spring weather we are having here in Calgary. If you're here, it's not uncommon to have these occasional snowy weathers here. So hopefully you guys are staying warm. But yeah, it was a pleasure to have everyone here, and thank you for joining us for the Lunch and Learn for March. Feel free to connect with the grad college. The links are available at the bottom here for any participants that aren't a part of the grad college as it is. And any information for, just you can find any information about our future Lunch and Learns on the website as well as on the social media that we have. So we hope to see everyone again next month when we have our April Lunch and Learn. Thank you so much, and we'll see you soon.